Today's speaker is Dr. Ashwini Hariharan from the lab of Dr. Thomas Longden at the University of Maryland in Baltimore. She will be talking about Pericyte electro-metabolic signaling controlling blood flow in the brain. A little bit introduction about Ashwini. Ashwini finished her bachelor degree in pharmaceutical sciences and then her master's in pharmacy as well at the University of Mumbai in India before going to New Zealand for her PhD at the University of Otago, where she worked on endothelial nitrocyte synthase and neovascular coupling. And then she saw the light when she started looking at ion channels when she started her PhD when she started her postdoc with Tom Longden at the University of Maryland in Baltimore. Since she started, she has been a postdoc for about 4 years now, I guess, and she has been doing great work. She published kind of a beautiful paper in Sur Reports a couple of years ago, and I think she will be talking about her ongoing research. Ashwini has been kind of presented multiple and several occasions over the years, presentations and talks and posters, and she's a recipient of a postdoctoral fellowship from the American Heart Association, and she has been actively involved with American Heart Association activities at different levels. With that, without any further delay, I will leave the floor to Ashwini. Thank you so much for accepting our invitation and looking forward to your talk.
Thank you. Thank you so much, Osama, for that introduction. And thanks to everyone for joining my talk today. So as you said, I'm a postdoc with Tom Longden at the University of Maryland. I'm in my 5th year now. And I've been working a lot on these cells called pericytes in the brain. They're a type of cell in the blood vessels of the brain. So I'll first talk about how these cells control blood flow in the brain. And the second part of my talk, I'll present some preliminary data on how the mechanism by which pericytes control blood flow is disrupted in aging and Alzheimer's disease. So I'll first talk about how important blood vessels are to maintaining brain health and function. As you can see in this image here of a cleared mouse brain, the brain is really well supplied with a dense network of blood vessels. It's almost 400 miles in length. And why this is important is the brain consumes a lot of energy on a day-to-day basis. You've probably heard of this factoid that the brain consumes almost 20% of our daily energy requirements. But the brain also has very poor ability to store energy. So it really relies on the blood vessels to supply neurons with glucose and oxygen in the blood. So what happens is when neurons are active, there is a regional increase in blood flow, which brings with it more glucose and more oxygen that then the neurons can use to make ATP that then sustain their activity. This is what we call functional hyperemia or neurovascular coupling. And the blood vessels are also really important to clear out waste. So they're really playing a lot of important roles to maintain brain health and function over time. So this neurovascular coupling, you see this phenomenon really beautifully illustrated in this focused ultrasound imaging of a rat's brain. What the researchers are doing is they're stimulating the whiskers of the rat and imaging blood flow in the contralateral somatosensory cortex. You can see that in the zoomed in version here. So as soon as they stimulate the whiskers of the rat, you see an immediate increase in blood flow in the somatosensory cortex. And this moment to moment energy and supply demand is what we call a neurovascular coupling. And the blood vessels in the brain play a really important role in doing this. So in our lab, we're really interested in the mechanisms that the cells of the blood vessels use to detect neuronal activity and respond with an increase in blood flow. So to understand these mechanisms, first we can look at the architecture of the brain and how these blood vessels are organized. So on the surface, you have these peel arteries that course along the surface of the brain, and at different points, they dive almost at right angles into the brain parenchyma to form what are called penetrating arterioles or PAs. And these PAs at different points form a really, really dense network of capillaries. So the peel arteries and the penetrating arterioles are covered by these ring-like smooth muscle cells. And every time the smooth muscle branches to a capillary, the smooth muscle cell converts to what are called pericytes. So the first branching point of a smooth muscle to a capillary is called the first order capillary, and that then branches to form two second order capillaries, so on and so forth. So the first few branches of these capillaries around first to fourth order are covered by what are called contractile pericytes. So they're also similar to smooth muscle cells in that they can actively contract and dilate the capillaries under them because of their expression of smooth muscle actin. And around the fifth order and above are these pericytes form a thin strand like morphology. So they're called thin strand pericytes. These are what are the cell types that I will be talking about today. And these cells, unlike the smooth muscle cells and contractile pericytes, don't express a lot of smooth muscle actin. So they instead rely on their ion channels and their electrical signaling to control blood flow in the brain. And this really, our thinking of how this happens is based on some of Tom's postdoctoral work, which basically found that this capillary network in the brain forms essentially a long electrical cable that detects neuronal activity and then responds by electrical signaling to increase blood flow. So, what happens here is these capillary endothelial cells are connected to each other by tight junctions, and they all express very high levels of a strong inward rectifier potassium channel, also called the KIR 2.1. And these channels, they're able to detect increases in extracellular potassium, and this causes them to open, and that hyperpolarizes the cell. So when there's neuronal activity, there is an increase in extracellular potassium in the vicinity of this endothelial cell, which causes the KIR channel in the endothelial cell to open. That hyperpolarizes the endothelial cell. And this hyperpolarization is then passed through these tight junctions to the next endothelial cell and the next, and so on and so forth, all along the capillary bed really rapidly till it reaches this smooth muscle or the penetrating arteriole. And here, the endothelial cell passes this hyperpolarization through a myoendothelial junction to the smooth muscle cell that's wrapping around it. And that hyperpolarization shuts down the voltage dependent calcium channels in the smooth muscle, which causes the smooth muscle to relax. That dilates this big arteriole, and that lets in a lot of red blood cells, causing vasodilation into the capillary bed, which then brings with it more glucose, more oxygen that is then taken up by these neurons, and that satisfies their energy requirements. So this is how this electrical retrograde electrical signaling works in the brain. And since our cells of interest, the thin strand pericytes, are located really very well positioned between these neurons and the endothelial cells, we started thinking about how these pericytes might fit into this idea of the capillary electrical signaling. As you can see in this image here, just to go back to the morphology of these vascular structures, you have the penetrating arteriole and the first few branches of the capillary network here that are covered by contractile neural cells. And then deep in the capillary network here, you have our cells of interest that are thin strand pericytes. So thin strand pericytes have this bump on a log morphology. They have the cell body here, and then they extend these really long, thin processes that cover almost 90% of the vasculature. So
we started thinking about how these cells might control blood flow in the brain, because unlike these contractile cells upstream, they don't express high amounts of smooth muscle actin. So that's why we started to wonder how then might they control blood flow in the brain. So to try and understand that, we looked at this single cell RNA-seq database that was generated by colleagues in Uppsala University in Sweden. So we looked at the molecular signature of these thin strand pericytes, and surprisingly, we found that almost half of all of the ion channels that they expressed was made up of the ATP sensitive potassium channel. So these KTP channels have two subunits. They have the inward rectifier potassium, KIR 6.1, and the sulfonylurea receptor 2, which is encoded by APCC9. So as the name suggests, these channels are ATP sensitive. So they work to couple cellular energy state to membrane potential changes. And you're probably familiar with KTP channels in the pancreatic literature, where they have the KIR 6.2 and SIR1 isoform, where they couple glucose levels or ATP levels to insulin secretion. So in the vascular isoform, this was a structure that was released in 2021. You can see that the KIR 6.1 isoform of the channel has these two nucleotide binding domains. So these nucleotide binding domains, they can bind ATP to keep the channels in a closed configuration. And when there is a decrease in ATP or an increase in ADP that binds to magnesium, this then opens the channel to hyperpolarize the membrane. So this gave rise to our central hypothesis, which is that thin strand pericytes are acting as metabolic sentinels. So this is where thin strand pericytes are located, deep in the capillary bed, and they exist in very close contact with the endothelial cells underneath. And they also form PEG socket junctions with these endothelial cells, which are thought to be sites of gap junction coupling. So we thought about the retrograde electrical signaling and the fact that these thin strand pericytes express KTP channels. And then we came up with the hypothesis that maybe they're continually monitoring energy substrate availability. So if there is a decrease in local glucose around this pericyte, maybe due to neuronal activity, it can cause a decrease in the ATP inside the pericyte, which would then activate the KTP channel to hyperpolarize the membrane, which then gets passed on to the endothelium underneath by PEG socket or gap junctions. And that engages the retrograde electrical signaling along the capillary network to rapidly dilate this arteriole upstream, which then increases blood flow into the capillary bed, brings with it more glucose and oxygen to protect neuronal health. So before we look at data that support this hypothesis, I just want to introduce the main technique that I've used throughout this project, which is in vivo multiphoton imaging. So to do this, we use a 2-photon laser scanning microscope. We have anesthetized mice that we inject with a dextran dye. So this dye stains the plasma, and the red blood cells can then be seen as black spots or black spaces in the capillaries. So we can get two main measures out of this. One is when we do Z projections of these volume acquisitions, we can get information about the diameter of the vessel. So you can see the penetrating arteriole here and the capillary network here. So we can measure the diameter of the vessels in response to drug application. And the other thing we can do is use line scanning to generate these chymographs that gives us the information on how these red blood cells travel as a factor of time. So this gives us information on blood flow. So the faster the red blood cells flow through this capillary network, it means the blood flow is higher, and so on and so forth. So the first experiment here was just to see if we can activate KTP channel in vivo by putting on a KTP activator, pinacetyl, just on the brain surface, and then measure the diameter of the vessels in response to that if we see if there is an increase in blood flow or an increase in vessel diameter. So when we do this maneuver, we do see that the penetrating arterioles and the first few branches of the capillaries are significantly dilated on opening KTP channels. And when we measure red blood cell flux deeper in the capillary bed, we see that blood flow in the capillary bed is also increased. So then we went on to see if pericytes actually express KTP channels, and that dilatory effect that we saw is actually coming from pericytes or from somewhere else. So we isolated microvessels from the brain of a mouse that expresses a red fluorophore in pericytes and smooth muscles. So when we when we mount these vessels, we can see our pericytes as red cells. And we use very high resistance sharp microelectrodes to impale these pericytes, which gives us a readout of the membrane potential. So here you can see that the normal baseline membrane potential is anywhere between -35 to -40. And when we put on pinacidil, these cells are hyperpolarized significantly. And when we block KTP channels with glibenclamide, that hyperpolarization effect is lost. So now that we've established that activating KTP channels can cause an increase in blood flow in the brain, and that pericytes express KTP channels, we wanted to investigate this electrical signaling mechanism in vivo. So to do this, we again use the pericyte fluorescent mouse. We inject a green dextrin dye in the blood plasma. And here we can identify all the vascular structures we are interested in to investigate that electrical signaling mechanism. So you can see the penetrating arteriole here, and it gives rise to the first, second, and so on, the capillary branches. And these are covered by the contractile pericytes. And deeper into the capillary bed, you can see our thin strand pericytes. So we identify our cells of interest, and then we maneuver this pipette containing a red dye along with pinacidil, which is the KTP channel opener, and we selectively apply pinacidil just on this cell. So you can see it in that panel here. Instead of applying a drug all over the brain surface, we can very selectively activate the KTP channels in just one cell. And after doing this, what we're getting out of this experiment is measuring the diameter of the arteriole here to see if that electrical signal is traveling upstream to cause a dilation. And we're also measuring blood flow down here to see if that increases red blood cell flux deeper in the capillary bed. So you'll see that in this movie here, this is a typical imaging volume. You see that's our penetrating arteriole where we record, and deeper around 180 microns away, we have our pinacidil, have our pericyte of interest where we're applying pinacidil. So when we do this and we apply pinacidil, we can immediately see an increase in the diameter of the arteriole. We can just run through this movie again. This is our recording side, which is the arteriole. This is the ejection side where we apply pinacidil. So we're measuring at the arteriole here, and now you see the dilation. So this is consistent with pericytes, KTP channel signaling upstream to increase blood flow. And this is the data presented here. You'll see after we do that pinacidil application on the pericyte, we can see a significant increase in arteriolar diameter, a significant increase in blood flow downstream. And to see if this engages the endothelial KIR or KIR 2.1 signaling mechanism, we blocked endothelial KIR channels with barium, and you can see that in the purple graphs here that eliminates the barium response, eliminates the pinacidil response. And we also applied pinacidil on a segment of capillary that doesn't have a pericyte soma. You can see that in this image here, the nearest pericyte soma is far away from where we're actually applying the drug. And again, when we do this, we lose the dilation and the blood flow effects. So these data really show that the locus of KTP channel activity lies in thin strand pericytes. And the final experiment in this series was to do the same experiment, but knock out the KIR channels from pericytes. So we use this Cree-Flox mouse that expresses a mutant KIR channel. And this is tagged to GFP. So when we give these mice tamoxifen, we see a loss of the GFP signal. And to really identify cells that don't have the KIR channel, and to identify cells that lose this GFP signal in vivo, we use this dye called mutatrace, which specifically stains pericytes, but only in vivo. So when we do this, we first look at a Z-stack of the mouse, and then we put on mutatrace to identify cells that will first dark and then appear as green spots. So these cells really show us cells that don't have an active KIR channel or an active KTP channel. So we can selectively target them with pinacidil. And when we do that, we see that, again, we lose that upstream arteriolar dilation and red blood cell flux. And then we repeat this experiment in the controls. We did 2 different controls here, a pre-control, which doesn't have the KIR knockout and was given tamoxifen, and a vehicle control, which had the KIR knockout, but was given vehicle. And in both these groups, again, we see that the KTP response is retained. So now that we've established that KTP channels are located in thin-strand pericytes and activating them can cause increases in blood flow and penetrating arterial dilation, we started thinking about what would physiologically activate these channels, because there's no pinacidil in our brains. So we thought about, okay, how do we manipulate energy substrate availability, and how do we see if this activates KTP channels? So we looked at 2 different strategies here. First was to see if we can change extracellular glucose, and the second was to block glucose import into the brain. So if you look at this table here, we looked at a bunch of studies in the literature to see where or what kind of values exist for glucose concentration in the brain parenchyma. And there's quite a range here. It goes all the way from 0.35 to 2.3 millimolar, and it averages out to around 1.4 millimolar. And the second strategy here was to look at glucose transporters in the brain. So if you look at this immunofluorescence image here, the DS red, or the red cells, are the pericytes, and the green is anti-GLUT transporter 1 antibody. So the GLUT transporter 1 is really very heavily expressed in the blood-brain barrier, and it's I think one of the primary sources of glucose import from the blood into the brain. So first we manipulate extracellular glucose, and this was done in the ex vivo setup with impalements. So we isolate these DS red positive pericytes, and we change extracellular glucose concentration and then measure changes in membrane potential. So interestingly, we tried a whole bunch of glucose concentrations. I tried 4, 2, 1, and then we tried a bunch of concentrations below 1 millimolar. And really interestingly, the membrane potential is very close to baseline till 1 millimolar glucose. And as soon as we go below that, we see this hyperpolarization of the membrane, which was sensitive to glibenclamide. And you can see that in this trace here at 2 millimolar, the membrane is very close to baseline. And as we change to 0.25 millimolar, you see this hyperpolarization, which is then reversed by the application of glibenclamide that blocks KTP channels. So this graph really shows the summary data of all the different glucose concentrations that we tried. So you can see this sort of trending towards hyperpolarization as soon as we go below 1 millimolar glucose. And this effect was also blocked by glibenclamide. So when we plot this membrane potential and glucose relationship as a graph, we can see that it has a really sharp slope. And the EC50 of this is very, very close to 1 millimolar. It's around, it's 934 micromolar. So we call this the Pericyte energy switch, since it essentially works like a switch. As soon as glucose drops below a particular threshold, you see a rapid activation of KTP channels, which hyperpolarizes the membrane. So next, to do this in vivo, we looked at the GLUT1 or glucose transporter 1 blockade to block glucose import into the brain. We used this drug called BAY876, which is a very potent GLUT1 blocker. When we put this on cells, we see that it hyperpolarizes the membrane by activating KTP channels. And when we put this on the brain surface in vivo, we see that it does dilate the penetrating material and all the contractile capillaries here. So when we did this in the presence of glibenclamide, we see that the effect was blocked. It's the second bars here. And the third bars here show the same effect of BAY876, but in the KIR6.1 AAA mutant mice. So this brings us back to our central hypothesis, which is that pericytes are acting like metabolic sentinels. They're continuously monitoring energy substrate availability. When there is abundant glucose or when pericyte ATP levels are high, the KTP channels are closed. And when there's a drop in local glucose or a drop in glucose import into pericytes, this might change the ATP to ADP ratio within the cell, which then activates KTP channels to hyperpolarize the membrane and transmit this hyperpolarization upstream to cause a dilation and increase in blood flow. So now we'll move on to the second part of my talk, which is now that we know that these channels are playing an important role and pericytes are important for controlling blood flow in the brain, we were really interested in how this is, if it is even broken in Alzheimer's disease or in aging. So Alzheimer's disease, as we know, is characterized by progressive memory loss. It's also another factor that's very prevalent is glucose hypometabolism. So if you look at this panel here, these are some FDG PET images from one patient that was tracked from at 3 different age points, at 53 years old, at 56, and at 58 years old. In this upper panel here, you can see the FDG uptake. So the red indicates high uptake and yellow and green indicate low uptake. So as this patient was tracked through their life, their MMSE score, which is a cognitive test, got lower and the glucose uptake was also lower. And they also compared the different areas of glucose utilization with control brains or control patients. And they found that at these older ages, there are these higher spots of decreased glucose utilization or glucose hypometabolism in this patient. So this is just one example of what is very well documented in AD, which is problems in utilizing glucose and metabolic dysfunction. As well as that, there are profound cerebrovascular abnormalities, both morphologically as well as in physiological function. And there are also alterations in brain blood flow. And you can see that here as well. This was a paper which looked at the biomarkers for different factors such as vascular function, amyloid beta, CSF tau, et cetera, and they've sort of attracted with age and the onset of diseases, which is late onset Alzheimer's disease or early mild cognitive impairment. And you can see here that vascular dysfunction is emerging really early on before the cognitive symptoms really appear. And this is important because sporadic or late onset Alzheimer's accounts for a majority of all Alzheimer's cases. It's over 95% of all the cases. And it also often precedes amyloid beta deposition and functional impairment. So a big part of my project now is to really understand if at all, pericytes are contributing to this vascular dysfunction that happens with aging and in Alzheimer's. So this is some preliminary data that I've been working on over the last few months. So if you remember from the previous slides, we had this effect of blocking glucose import that causes a dilation of the vessels, which is mediated through KTP channels. So we wanted to look at how this is affected in older mice. So we used 12 to 15-month-old mice, which are late middle-aged. And we used this mouse line, which is a 5X FAD, which expresses a lot of familial AD mutations. So we compare the wild type with their transgenic littermates. So the wild types don't have any AD mutations, and the transgenics have the AD mutations. Surprisingly enough, we found that in both of these groups, the diameter of both the arterial as well as the capillary was unchanged to BA876. So basically, these vessels did not respond to blocking glucose import, which suggests that this pericyte energy switch is defunct in the aging brain. And we also looked at this ex vivo. So instead of making more fluorescent mice, we used this AAV approach, which is the AAV BR1, that very specifically targets the brain endothelium. So by using this, we injected AAV BR1 GFP to the 5X FAD mice. And here you can see the endothelium is green. And we then identified pericytes under the bright field microscope as colorless cells. So when we impale these pericytes here, again, we see that they're not responsive to BA876 at all. And these are, again, older mice, 12 to 15 months old. And they lose the hyperpolarization to BA876 that we see in younger mice, which again supports the in vivo data that this energy switch is broken down with aging. So then we were kind of confused because we were initially expecting to see some effect of, the AD mutations, and we thought the wild type mice would be fine. But since we saw that at this 12 to 15-month age point, in both the groups, this mechanism was broken, we then moved to a middle age group, which is 6 to 8 months old. And here, interestingly enough, only the transgenic mice performed worse and the wild type mice were fine. So there is this progression from 2 to 6 to 12, 15 months old, where the wild type mice are fine till we see them at 12 to 15 months old. But in the presence of Alzheimer's mutations, we see that this breakdown is sort of earlier at 6 to 8 months old. And we see this also in the membrane potential changes. So then we started thinking about the mechanisms. Why is this happening? What causes KATP channels in pericytes to lose their sensitivity to changes in energy substrate levels? This is when we started thinking about because KATP channels are in the membrane, what else can be in the membrane? Is it something to do with how these channels are organized? That goes wrong with aging, that causes them to lose the metabolic sensitivity. This is when we came across caveolae, which are these flask-shaped invaginations in the membrane. They are really rich in lipids, phospholipids, sphingolipids, as well as cholesterol. And they also have high levels of this protein called caveolin. And they're very easily visualized in electron micrographs. As you can see here, they have these flask-shaped membrane invaginations. This is in the endothelial cell. So another function of caveolae is they can sort of sequester ion channels and their signaling complexes in the membrane. So this is just an example. Here you can see the ion channel. And so we started thinking, well, maybe in the aging brain, if there is an increase in this cholesterol or an increase in caveolin that sort of interferes with how this ion channel is interacting with maybe glucose transporters or other metabolic machinery that causes problems in the ion channel being able to detect changes in substrate levels. So maybe it cannot detect that ADP is going down, and that causes inactivation. And when we looked at the literature, it was interesting to see that caveolin-1 actually does interact with KTP channels. So in this paper, they co-expressed caveolin with the KATP channel, and they looked at the sensitivity of the channel to ADP. So when they do this in the absence of caveolin-1, they see that the channel or the current is higher at lower ADP concentrations. The channel is really more sensitive to small ADP changes. But when they express caveolin-1 along with these cells, this concentration response is shifted rightward. So the channels are less sensitive to changes in ADP. So there seems to be some interaction between caveolae and KTP channels, which can affect how these channels are sensing ADP changes. Then there's also some evidence, admittedly not a lot of evidence, but to suggest that caveolin can increase with age and in Alzheimer's disease. These are both from brain tissue. The AD is from, they're both from human brain tissue, actually. And you can see here that in older people, there's an increase in caveolin-1 protein expression, as well as in Alzheimer's disease patients, there's an increase in caveolin-1 protein expression. So this was a really interesting sort of tool that we wanted to manipulate. So there's not a lot of great pharmacology to inhibit caveolin specifically, but because these are so rich in cholesterol, we thought we would use this drug called methyl beta cyclodextrin. And in the literature, it's been used quite a bit to inhibit caveolae or disrupt caveolae. So when we do this in vivo, when we add methyl beta cyclodextrin along with our glucose transporter blocker, interestingly enough, in older mice, we see that we can restore the vasodilation effects that were absent without the methyl beta cyclodextrin. So something about inhibiting caveolae is actually restoring the sensitivity of the KTP channels, because this effect was again inhibited by glibenclamide. So interestingly enough, it seems like this caveolin KTP interaction, if we disrupt it, we can restore the metabolic sensitivity in older mice. So to just bring this all together, I'll talk about some directions that we're taking this now. The first is we're really interested in this sort of molecular interacting partners of KTP channels. What does the Pericyte KTP metabolome really look like? What kind of molecular partners do interact with these cells that help them detect these energy changes, whether it's glycolysis or glucose transporters or oxidative phosphorylation or something else, and how this changes with age or in AD. The second project here is to look at whether we can harness some pharmacology to inhibit cholesterol and caveolae in pericytes. So this is some very preliminary data using a statin, which has been shown to disrupt caveolae abundance in the literature. And when we use this in vivo, we do see some effects of restoring the B876 hyperemia. And this is also sensitive to glibenclamide. So this is a promising lead that we have. And the final part of the project is to do some ultrastructural investigation. So these are just some representative EM images or transmission EM images that you can see the red blood cell here and the endothelial cell. And in the pericyte here, you can see, very interestingly, the caveolae is sitting right next to the pericyte mitochondria. So we want to dive a little bit deeper into these sort of ultrastructural investigation of pericytes in both aging and AD contexts to probe this interaction between caveolae and pericytes and see if that underlies the dysfunction that we see. So I'll end there. I just want to thank the rest of our group, especially Tom, and also our collaborators, both at the University of Maryland, the EM Core, as well as our collaborators in Uppsala and the American Heart Association for giving me my fellowship, which is nice. And I'll take questions now.
Thank you very much, Ashwini. That's a great talk, and we have already a few questions coming in, and I anticipate more questions coming. So the first question that I can read here from Giovanni Jizyan, and I apologize if I didn't pronounce the name correctly. Do the pericytes coupling in those sites show any expression changes of connexins, since those channels could respond to glucose levels and may act as conduits for both electrical and molecular signals?
Sorry, can you repeat that one more time?
Sure. Yeah. Do pericytes kind of show change in expression of connexins under different conditions, since those channels could respond to changes in glucose levels, and therefore they can kind of change signaling at the electrical level and molecular level?
That's a really good question. We haven't looked at changes in the expression of connexins in pericytes yet. We have thought about looking at gap junction coupling, and that's again something that we can do with EM, is to look at PEG socket junctions that in the disease context as well as in younger mice, and that should give us an idea of whether or not aging or Alzheimer's can impact how pericytes and endothelial cells can interact. So if there is a lesser abundance of PEG socket junctions, that would indicate that the connectivity is lost, or they're not able to communicate as efficiently.
I'm just following up on the same question, and that's kind of my personal question. Is it known which connexins are kind of coupling pericytes into T cells?
So from the RNA-seq database that we mined, the two connexins that jumped out were connexin 37 and connexin 43. We haven't really done any further analysis to confirm this, but those were the most highly expressed in pericytes and endothelial cells. So that really is very suggestive that these are the connexins that form the PEG socket junctions.
Sounds good. The next question from Osama Saidi is, can you comment on the relationship between diabetes and pericyte function, given that your work implicates metabolism and glucose sensitivity in their function?
That is a very good question. I have personally been very interested in insulin signaling in pericytes in the brain. I couldn't comment directly about whether or not pericytes are responsive or play a role in blood flow control in diabetes. We do know that blood flow in the brain is impaired in diabetes. In fact, so pericytes, interestingly enough, do express all the elements that are necessary for insulin function, like insulin signaling. So they have the insulin responsive glucose transporter. They express the insulin receptors as well. So this would be a really interesting sort of something interesting to study, but I haven't looked at it personally.
Sure. The next question from Ashinafi Bitri. Fantastic talk, Ashwini. Greetings from Australia. Question number 1 is 1 millimolar extracellular glucose seems low compared to physiological concentration in the blood. What is the physiological kind of concentration, kind of effective concentration levels of glucose? And the second question is, if both barium and glibenclamide inhibit the effect of phenazidol, I wonder how you determined if KATP and not KIR 2.1 is primarily involved.
Okay, so I'll answer the first question, which was I forget what the first question.
The concentration of glucose, 1 millimolar versus what is physiological. Yeah.
So in the blood, I think glucose is upwards of 5 millimolar. I think it's depending on fasting state, it can be anywhere between 5 to 6. In the brain, and this is very tightly controlled because we don't want too much glucose lying around in the brain parenchyma, which can, obviously cause problems for neuronal function. This is really, really tightly controlled. This concentration gradient from the blood to brain glucose is really important to maintain brain glucose as at this low level. So you constantly have glucose only coming in one way, right? You don't want glucose going out of the brain into the blood. So this is really important to consider. And the second question, I guess, was
That barium and pinacetyl, both of that, and glibenclamide, sorry, they both inhibited the effects of pinacetyl.
right. So this is why we did the selective application of pinacetyl on the pericyte in a few different contexts. So we've done the application of pinacetyl in the presence of barium to block just the endothelial KIR channels. And we did the application of pinacetyl in the KIR AAA knockout that has no barium and no glibenclamide. So that's how we can sort of distinguish that the contribution of the endothelial KIR 2.1 channels from the pericyte KTP channels.
Sure. The next question from Sinri Lim. Since cholesterol is implicated in cellular membrane fluidity, can you comment on how stripping this may affect your responses? And that methyl beta cyclodextrin may also strip other lipids. So do you have some other candidates in mind?
Yeah, that's a really good question. This is why we're really looking to use some other drugs to target this mechanism, because we don't want to really disrupt the membrane integrity. We just want to restore the sensitivity of the KATP channels. So one promising candidate that we are trying now is the statin that I spoke about very briefly. In this case, we will probably want to titrate the dose so as to just have a moderate effect. We don't want to really strip the membrane or disrupt the membrane integrity. We're also planning to do some ex vivo experiments where we can really look at the membranes in the different disease groups, just to see how that is sort of changed with aging and as well as with Alzheimer's.
The next question from Maria Notterman is, do only Pericyte soma sense local glucose availability? And I think this might be related to one of the experiments you showed about kind of the application on kind of on a non-soma location of a capillary.
Yeah, I mean, I think this is hard to address in vivo. Just because, like, even when we try to do the application really locally, you can sort of do it just on the cell body and not on the soma, right? So at least from the experiments that we've done in vivo, it would appear that there may be more ion channels in the soma, so they're more sort of sensitive to changes in energy levels. But I wouldn't dismiss that the processes are not sensing glucose as well.
Sure. Next question from Colin Nichols. Great talk. I guess I didn't really get your take on how the signal passes upstream. Do you think the pericyte projections communicate directly through gap junctions to the contractile cells, or is the connection via intervening endothelial cells?
So I think, so from our data, it would appear that the pericytes are communicating to the endothelial cells that they are closely opposed to via gap junctions. And that hyperpolarization that's passed to the endothelial cell then engages the KIR signaling mechanism, that's the retrograde electrical signaling, to then pass the signaling upstream to the arteriole.
Sure. The last question we have now is from Ibrahim Saleh, saying that what kind of imaging modalities did you use to image the pericytes, and what kind of stains do you think is the best for specifically staining for pericytes?
That's also a good question. So for our in vivo experiments, we use fluorescent mice. We've used in this talk, I've only used the DS-RED mice, which express DS-RED under the NG2 promoter. But now we've moved on to a PD-GFR beta, which is the platelet-derived growth factor receptor beta promoter to express TD tomato, which is, again, both these lines are specific for mural cells, so they do stain smooth muscle and contractile pericytes as well. Right now, we don't have anything that's very specific for just thin strand pericytes and not contractile pericytes. So that's been one of the challenges. But usually we can sort of distinguish them based on morphology. So the thin strand pericytes, you see these really long processes and you see them deeper in the capillary bed. and the contractile pericytes have a very different morphology. With respect to stains ex vivo, for contractile pericytes, hydrazide stains work pretty well, but for thin strand pericytes, we do have to rely on fluorescent mice.
Sure. We have two more questions. One from Artem Kondatsky. Thanks a lot for this colorful presentation. I learned something new today. So it seems that vascular smooth muscle cells regulate blood flow in bigger vessels and pericytes in capillaries. And both of them use KATP channels to regulate vascular tone, contractility, and consequently blood flow. Minoxidil, which is a KATP opener, is used for hypertension. Could it be used as a brain anti-aging tool?
That is a great question. I think the only problem I would foresee with a broad spectrum KATP opener that is not specific for a vascular or a pancreatic isoform is that the KIR 6.2 channel or the pancreatic isoform is expressed in neurons in the brain. So that might have some off-target effects that you probably don't want. I think there was a paper recently where they've developed a very specific vascular KATP opener. I cannot remember off the top of my head, but something like that would probably be a better strategy, I think, to prevent hypertension.
Yeah, the last question we have now from Colin Nichols, again, one follow-up question. I missed what you did with barium, but did it block the upstream transmission from pinacetyl?
Yes, it did. So when we do the specific application of pinacetyl, so we do this as a paired experiment where we put on pinacetyl as is, we measure the upstream arteriolar diameter, then we put on barium and repeat that pinacetyl application, and we see that the upstream dilation is eliminated.
Yeah, I think that's all, and we are right on time. And Colin says, "Cool, thank you." So thank you again, Ashwini, for a great talk and very engaged kind of questions and answers. Thank you very much for accepting the invite. Really looking forward to seeing more about your work. Yeah, thank you very much.
Yeah. Thanks.
Thank you.
Thanks, everyone.